In the early phase of structure-based drug design, researchers prepare a target protein and look for small molecule ligands that would specifically bind to the target protein. The ligands are usually collected from a chemical database. Molecular docking is a preferred method to predict how the ligands interact with the target protein in order to identify active compounds from the chemical database. However, it is well known that docking scoring functions often fail to reliably discriminate real binders from non-binders. Therefore, an alternative or complementary filtering method is needed to improve docking results and more reliably discriminate binders from non-binders. To solve this problem, we have recently shown that high-throughput molecular dynamics simulations can be used to improve docking results of protein-ligand complex structures. Specifically, we evaluated 560 protein-ligand structures consisting of 280 active sites and 280 decoy ligands, and showed 22% improvement in area under the curve in its ability to discriminate active compounds from the decoy compounds. This video demo will demonstrate the general functionality of high-throughput simulator in CHARMM-GUI. HTS automatically prepares simulation systems of multiple protein-ligand complex structures in a high-throughput manner to facilitate structure-based drug design. On the front page of HTS, we can upload all our protein-ligand complex structures by dragging them into the upload area. Note that HTS allows variations of protein structures in the ligand binding site that could result from flexible docking. In this example, we will build 5 systems of protein-ligand complex in solution with the same protein structure and 5 different ligands. The protein is a tyrosine protein kinase LCK with PDB ID 20F2. This kinase is expressed in T cells and plays a critical role in T cell receptor signaling. Thus, an effective inhibitor can be a potential autoimmune agent. Using VINA, we already docked 5 ligands into the binding site of the protein and named the protein ligand structures complex 1 through 5. These 5 structures can be downloaded by clicking the link in this video's description. Click Next to read the structures. After CHARMM-GUI reads our protein and 5 ligands, click Next to go to PDB manipulation options. If we want to modify the protein structure, this is where we would do it. To learn how to use the structure manipulation options, please see the video demos for PDB Reader and Manipulator. In this case, we do not need to modify anything, so click Next to generate a CHARMM-compatible PDB file, as well as ligand force field parameters. Don't forget to save your job ID, as you will need this number if you want to retry any steps with Job Retriever. Your job IDs are also available in your user profile. When it finishes, you should view the structure to ensure that it was read correctly. Our protein ligand structures are listed in the table on this page, with their ligand IDs and ligand force field parameterization. HTS offers 3 force field options, including CGenFF, GAF2, and OpenFF. When a particular ligand is successfully parameterized, a green checkbox is shown. If the ligand fails to parameterize, a red X mark is shown instead. In this case, all ligands are successfully parameterized. In addition, HTS allows structural modification of any of our ligands. This can be useful to modify our ligand when a particular ligand cannot be parameterized by our choice of ligand force field. If we want to change the force field type for our ligands, we can simply click one of the 3 selections above the table. In this case, we will choose CGenFF. Click Next to solvate the systems. On this page, we define the water box size, type of ions, and ion placing method. Click Next to set up periodic boundary conditions. On this page, the solution systems box is replicated for each protein ligand structure using the system with the biggest ligand. Water molecules that overlap with protein, ligand, or ions are removed. Each system is neutralized by deleting ions depending on the charges of a specific ligand in a system. On this page, we can generate a variety of MD simulation input options including NAMD, GROMACS, AMBER, OpenMM, Genesis, Desmond, LAMMPS, and Tinker. In addition, hydrogen mass repartitioning is also supported. Once you've selected your desired simulation programs, click Next to generate equilibration and production inputs for each system. On the last page, we can download the input files for running MD simulations of our protein ligand complex structures.